Thank you, Kevin, and good morning, everybody. So this morning's topic is hot topics. And this is traditionally a symposium where we put in a kind of eclectic group of folks that are doing interesting stuff that's interesting to everybody. So our first speaker this morning is going to be Dr. Michael Snyder. And Dr. Michael Snyder is a person who has been doing a lot of work in not everything from full genomics to how one monitors their own health and physiology, and how we can apply this to in training our bodies to be more healthy in adverse environments like, say, going to space. So I'm going to turn it over to Mike Snyder and let him tell us about his work.
Thank you. Okay, great. Well, it's really terrific to be here and tell you about some of the things we're up to. We basically do a lot with big data, and that's the theme I'm going to hit you with, and you'll see how it applies to space. I hadn't realized Chris had talked earlier, but I'll give you my flavor on some of the things we're doing with twins. So let's see if we can get going here. All right, there we go. So we're really focused on trying to keep people healthy, and that's very different, I would argue, from the way medicine is currently practiced. That is to say, in fact, I think the healthcare system, if you ask me, is totally broken, because usually people go to the doctor when they're ill. They rarely go when they're healthy. And so what we're trying to do is try and focus on keeping people healthy. So what does it mean to be healthy? Well, your health is influenced by many, many different things. It's influenced by your DNA. Let's see if I get there we go. So your genome, but also many other things that you encounter. So your exposures, your environmental exposures, your lifestyle, meaning the food, you eat, exercise, stress. We know all these things impact your health, but we're now at an age where it's possible to actually measure all these things. Some of them clunky, that is, measuring food is still doable, but it's a little bit clunky, but some are automatic. That is to say, we can measure people's exercise routinely, whether you're trying or not. And that's because there's a revolution going on in technologies that let us measure many things. So even when we're not measuring the various things we're exposed to, we can still measure their effects by measuring our biochemical profiles. So a number of years ago, we set up some, oh, sorry, I jumped ahead. We do this using combinations technologies. One, as everyone here is very familiar with, the revolution of DNA sequencing. We now sequence a genome for under $1,000, about $780 what we pay for a genome. The interpretation is much more. But there's a revolution going on in mass spectrometry, you may realize, where you can now measure thousands of features in people's blood and urine. And there's certainly a revolution going on in wearable devices. Just curious how many of you are wearing a smartwatch or some sort of wearable device there. Pretty good crowd. This is a high-techy bunch. Great. Okay, so anyway, we would argue all these measurements can be very, very powerful for following people's health. So a number of years ago, we set up something we call personal omics profiling, an integrated personal omics profile, or IPOP, where we do omics profiles, meaning we'll measure the genome, someone's DNA, once, but then longitudinally we'll measure many, many other components, mostly out of blood or out of urine. So we'll measure their epigenome, meaning their DNA methylome, their transcriptome. These are all out of peripheral blood monocyte cells, your immune cells. We'll follow proteome out of those cells in plasma using mass spectrometry. We'll follow cytokines. We've done a lot of work with metabolomics and lipidomics, so we've been optimizing that for the last 6 years. We follow thousands of features, again, out of blood and urine. Microbiome, I'm sure you've heard about. We follow people's gut and nasal microbiome, and now we've been measuring tongue and skin. And on top of that, we have wearables on our folks, deep clinical testing, questionnaires, so on and so forth. So we do very, very deep profiling, and we do this longitudinally. We sample people roughly every 3 months when they're healthy. When an adverse event comes along, like a viral infection or maybe a colonoscopy, or even a fun occasion like a trip, we'll sample people much more frequently to see their biochemical changes that are occurring during these periods. And so far, we've been following roughly 109 people for around 6.5 years. One person, me, we've been following for 9.5 years. So we've been doing these deep profiles. And why are we doing this? We're trying to understand what it means to be healthy. How is it that you differ from the person sitting next to you? How does it change over time? What happens when you get ill? And perhaps most importantly, if we apply these advanced technologies to people, can we use them to better manage their health? And the answer is yes. So it turns out, just jumping ahead, after the first 3 years of study, we have 49 major health discoveries by putting this battery of advanced tests, following people very, very closely. And some of these things are pretty, I would argue, a big deal. They cover all kinds of areas: cardiovascular, hem-oncology, metabolic, and so on and so forth. Even picked up a case of Lyme disease. All these are picked up because we had these advanced technologies on people. So I'll walk you through some of these more in a minute, but we did pick up one case of early lymphoma, 2 people with pre-cancers, they're called, something called MGUS and smoldering myeloma, that with high frequency can convert to cancer. 2 people with very serious heart defects, I'll go into one in a minute, and so on and so forth. So we can, again, find these different things. And the key was that no one technology actually found all these things. It took the combination to be able to do this. So a lot of this was actually picked up from 12 discoveries, and actually 13 if you count pharmacogenomics, was picked up through people's genome. That is to say, we sequenced their genome and then we looked in detail at what they might be at risk for. And as an example, most of you are probably familiar with the BRCA mutation. One individual had a BRCA mutation. It was a male, but he still has an 8% chance of getting breast cancer, so he'll get screened more. And not only that, his sister, so he could alert his sisters about the possibility of they may have high risk as well. There are 3 other cases where there was actually very consequential information. There's an individual who had a mutation in this gene, which puts him at high risk for cardiomyopathy, a young guy. Turns out then he did a follow-up stress echo, and he's got a heart defect. And it's actually on drugs now. And this is all caught because we looked at his genome. This is an individual also, there are 2 people with this mutation, this SDHB gene, that puts you at high risk for certain kinds of cancer. One, again, fairly young person had this, and they did a whole body MRI. And it turns out they had early thyroid cancer, which was caught and then managed by surgery, and they were able to keep most of their thyroids. They don't need thyroid replacement therapy. So catching this information was very, very valuable for their health. We had 9 people who were diabetic. We're very interested in diabetes for reasons you'll hear in a minute. And 9 were thought to be diabetic coming into the study. One of them turns out, they were all thought to be type 2. One of them turns out has a mutation in this gene. They're not really type 2. They're a different kind of diabetes called MODY diabetes. And the way you treat a MODY diabetic is very different from a normal diabetic. They go on sulfonylureas, and this person's been on the wrong medication for years and has now shifted over and has a much more effective medication as a consequence. So we would argue this genome sequencing can be very powerful, even though, again, it's not the majority of people. It's actually 12 out of 70 that we first looked at have this. So 17% has something extremely informative in their genome. As I say, we did pick up other things. We had one individual who, it turns out, from imaging, had a large spleen. They also had elevated certain B-cell markers, and it turns out they actually have this early stage lymphoma long before they were symptomatic, and so it was caught early and treated. And we even have a candidate biomarker that tracks with when we caught the onset of lymphoma. That was here, so retrospectively we could look back to what markers track. This one's a cytokine that makes a lot of sense. We're going to see if this turns out to be a marker for a lymphoma in general. We had 2 people with precancers, I think I mentioned. These are picked up by what's called outlier analysis. This one individual, there are 1,000 samples analyzed. This one individual's 2 samples had way too much IgM compared to everyone else. So then we did follow-up tests, and sure enough, he's got way too many IgM producing cells, which is a precancer. 15% of the time that turns into aggressive cancer. And so knowing this, he'll get monitored more frequently. My own view is he should actually have his stem cells saved now while they're precancer, rather than wait till he gets cancer, and then you actually have to deplete the cancer ones, put them back. It makes a lot more sense to deplete the precancer cells, put them back, so you'd be low risk. But MDs thought that was a good idea, but nobody does that. We're not very geared towards preventative medicine. We're very geared towards trying to fix people after they're ill, which is bad. Okay, so sorry this keeps advancing. I must have a timer on somewhere. Anyway, 6 people had plaques in their arteries. I'm one of them. Most cases rather mild amount, but it's caught early, and this is picked up through imaging. And the recommendation here is you increase your statins. That was done for me, and now I can see my plaques are disappearing. So again, by collecting this information, it's very, very useful. Coming into the study, as I said, 9 people thought they're diabetic. A few people knew they were pre-diabetic. Well, it turns out 2 people were diabetic who had no idea. And 40-some% were pre-diabetic, most of whom did not know. In fact, 9 out of 10 pre-diabetics don't know they're pre-diabetic. I guarantee there's a lot of you sitting here right now who are pre-diabetic who don't know. And the best way to measure this, we would argue, is something called hemoglobin A1C, and also continuous glucose monitors are powerful. I was really happy to see the breakfast out there. It was not all muffins and cereal, because that stuff will just spike it'll basically kill you. It's worse than smoking, I think. Okay, so 80% of people spike the cornflakes in milk. Yeah, that's a very popular cereal. So as I say, that's worse than smoking, I'm sure. So anyway, during the course of the study, then, this is one of the main reasons we did this study. We wanted to see how people became diabetic. So a lot of people had diabetic values. 9 were officially classified as type 2 diabetic during the course of the study, and then more became pre-diabetic. And we can follow how they became diabetic. 2 of the 9 got there by gaining weight. The other 7 did not gain weight. 2 of them spiked their way in. This is one of them. This is actually me. Ooh, sorry, I think the timer will, pretty sure will come off later. But this is glucose in orange. That's hemoglobin A1C, a better measure for type 2 diabetes. It's a measurement of steady state glucose. And so I actually spiked my way in because I became diabetic range first after a nasty viral infection. And then this peak looks wimpy, but that's about 12 months of data. So I was high. I actually had to change my lifestyle. Getting a better pointer here, fantastic. Okay, so I actually changed, yeah, that's the powerful one. All right, so anyway, yeah, I changed my lifestyle, meaning I cut out all that cornflakes and milk I was telling you about before, and basically had been biking, doubled my biking, started running, brought it down to normal values, so boringly low that I stopped looking at it. And then someone else looked at it here and said, Mike, your hemoglobin A1C is 7.1. That's back in diabetic range. More than back, it's actually pretty high. So I'd stopped running, guess when? Right there, when it went up again. But I also had a viral infection, so it's statins, so it could be any of those. But the point is, it came back again, and I started running. I could bring it down, but I could never get it back to speed. So what I did instead was, I got it down to 5.7. This is several years, about 5 years of data, 4 years of data. And it just kept gradually creeping up. And so I'd been running about as much as I could, and you can't tell, but 3 years ago, I shifted to weightlifting here. And the idea was muscle mass is better for glucose homeostasis, and that failed miserably. It kept creeping up. Okay, so and in fact, it went up to 7.5, just right about there. And so I said, finally, all right, I got to take metformin, which is the #1 drug you take for type 2 diabetes. And guess what? I'm a metformin non-responder, which is about 20% of people. So in fact, as I say, it kept going up. I forgot to say, most other people don't spike. One other person spikes like me, and the others gradually got there. So what did I do? Well, I went back and looked at data. It's all about data. So what I discovered, this is complicated, but I looked at my insulin secretion. That was normal. Turns out I'm insulin sensitive. My cells respond to insulin just fine. So what's going on? Turns out I don't release insulin well from my pancreas. So I make insulin, my cells can respond, but it's not getting released out of the pancreas. And you can tell that because there's a very slow glucose disposition. I'm like at the bottom here, meaning I don't get rid of my glucose fast enough because I'm not getting my insulin out. So what's the solution? Well, I take actually something called a secretagogue. It's a prandial derivative called rapinolide, and that actually stimulates insulin release from your pancreas. And it works like a charm. I've driven my hemoglobin A1C in just a few months down to 6.3 from 7.5, and hopefully I'll keep it going. So the point out of this is actually knowing exactly what's wrong with you leads to the right prescription. This drug I'm taking, that's not a second-line drug. It's usually like 5th on the list. You usually work your way down the list, and I could zoom right in because I knew exactly what was wrong with me. And I would argue this is precision diabetes is the way precision medicine should operate in the future. We also, this is probably the most important slide here. We're all different. we all have different baselines, no matter what kind of measurement we do. This is using cytokines, metabolites, clinical labs, transcriptome. Even this one is different. Every dot here is a different person. Or sorry, every dot's a different visit. Every color is a different person. And so what you'll see is that they cluster by person, meaning they cluster by color. And this is, in my case, on the blue dots, that's 6 years of data. My dots still cluster near me, not near other people. So we're all different. What that means is if you get up off your seat, we took a swab, we'd know exactly who sat there if we had the right reference markers. That has other implications, of course. But the point is that we all have our personal signatures. They're robust. They don't change that much over time, except for some interesting aging markers we've seen. Even more importantly, it turns out if you undergo a perturbation, say you get a viral infection, colonoscopy, in this case, these are folks who did a weight gain, weight loss study. We're looking at their microbiome. We can compare them, cluster their samples, and you will still look more like you. There's some exceptions, but in general, you look more like you than the person sitting next to you, even if you get a viral infection. So your personal signatures are extremely robust. And so what that means is that, again, they're just so strong. This is why, in general, when people do studies, they feel they need thousands of people. And that's because the difference between disease and normal is smaller than the difference between people. So you need lots of people so you can catch that difference. Does that make sense? It's the most important conclusion to use, to know. And the reason this is a big deal then for following yourself, you can't miss that change if you know your basic health, your base health state. You see the shift between your disease state and your health if it's just you, as long as you got your healthy measurement. But it's hard to see the difference between your disease state if you're comparing you with everybody else in this room. Does that make sense? So you really want to get your healthy baseline and see that delta, and that's the way medicine has to move in the future. You can't miss that even on an N of 1. And we would argue that's, again, the most important thing to know. And this is shown here for microbiome. These are just 9 people. They all went through a weight gain, weight loss study where they gained 6.5 pounds on average and lost it. And when you cluster them by their microbiome, they cluster by the person, not by whether they gained or lost weight. Okay, I won't have time to get into this, but we're doing a ton with wearables these days. In fact, I use 8 of these devices every day. I have 3 smartwatches here. This ring's not really a ring, it's a sensor. They measure all kinds of very important things. So in fact, the average one takes about 250,000 measurements a day. So you can follow people's baseline, and again, you can see shifts. And I won't have time to go through this, but we actually think we can now tell when you're getting sick before you know it, because your heart rate goes up about 1.5 beats on average before you actually feel symptomatic. So we think we can now tell from this data. And that's how one of the ways I figured out my Lyme disease. We would also argue continuous glucose monitors are important too. So we've actually built now what we call personal health dashboards to be able to collect information from wearables, from your clinical labs, even your microbiome, you can collect and bring it to your smartphone. So we think this is going to be powerful. This is going to be your most important health instrument in the future, more important than your doctor, I would argue. Because you collect your information, just like your car dashboard, you can see what's going on. The goal is to have a health dashboard on your smartphone. And we, again, can collect all this wearable data, all kinds of data, and you can track what's going on with your health and put the power back to the person, because we think people can manage your health better than a physician who only has 15 minutes to look at you in a physician's office. We'd also argue that this is not meant to replace physicians. Meant to augment them, because you can share your data with your physician. And I guarantee, by the way, the resting heart rate you get from your smartwatch is better than anything you'll measure in a physician's office. So we've actually got the data for that. So my heart rate, when I go into a doctor's office, I don't get nervous because I've given blood 300 times in the last 3 years. And I don't get nervous, but my heart rate will vary 40 beats per minute, depending what's going on. Meaning if I rode my bike, drove, whatever, it's just all over the map. It's almost a worthless measurement, to be honest. But I can pull my resting heart rate off my smartwatch, get a much more accurate assessment of what's going on, and know A, whether I'm sick, B, whether I'm stressed, because I can see that value at a time, and it makes many, many more measurements. So I think, not for everything, but for some things, these measurements are gonna be more accurate than what a doctor does. And so you can share that information then with your physician, we would argue, and that will be very, very powerful. So we've been applying these. I hadn't realized Chris talked, so I'll give you my flavor on this. So we've taken this study and actually even taken it much further into space, so to speak. So a team of 10 groups, you probably heard from Chris, actually studied the twins. One went in space, one stayed on the ground as a control, so to speak. And we followed them, sampled them. Well, this is Scott when he was in space. This is Mark when he's on the ground. Sampled them when they're on the ground, several times in their space, and when they return. But the bottom line, as you probably heard, is we had all the same assays I mentioned before, plus more. We measured telomeres, many other things, and they're certainly, in terms of measurements, they're the most heavily measured folks, I would argue, certainly off the planet, and to some extent on the planet. Okay, and so what we saw is literally thousands of changes. These are just some of the assays that our group was involved in, plus cognition. So you can see there are dramatic shifts when people go into space, and they cover all kinds of different areas. And sorry, this came out blurred. But to put this in perspective, I think these are some of the major things we learned by following their transcriptomes, looking at things that shifted up. Most things, and that'll be a main theme, did return when Scott came back to Earth, but a few things did stay elevated. Immune response, DNA damage, even blood pressure and hypoxia, all increased when Scott was in space. And for the most part, they came back. The immune response actually stayed up a little bit, at least for 6 months. That was corroborated by looking at cytokines. So again, these are the things, each row is a different cytokine. You can see this would be, these over here are Scott, and you can see they burst, they go up when he's in space, and for the most part, they come back. There are a few things that can go the other direction. Lipids are very, very interesting, and we don't know enough about these, I would argue. There's literally thousands of lipids, and the little bit we know about, we always think of they're boring membrane proteins, but they're actually incredibly interesting signaling molecules as well, and there's many roles yet to be discovered. And what we're discovering in this case, there are clearly shifts for Scott when he went into space. For the most part, again, they return, but omega-3s, which are thought to make you smarter, you get out of your fish, those things did shift. I'm not sure that he got smarter when he was in space. But anyway, the point is you can follow all these things. Same with the omega-6s. Those are generally not good things. There's something called arachidonic acid. That turns out to be an incredibly interesting signaling molecule. We could see shift in Scott. And actually in other people, these other folks I mentioned, when they're actually undergoing certain immunological stress. It's quite an interesting marker, we think. This is from Brina Rana's lab. She was looking at mitochondrial shifts. We could see those in space as well. This is, I want to flip through these because we have lots of time. There are interesting cardiovascular changes. We can actually pick these up as well in the healthy folks on Earth. So yeah, this one ratio of ApoB to A is a good marker for cardiovascular health. And we actually can see shifts on Scott when he's in space. And there are other markers as well, collagen, that we can see change. So the point out of all this is we do see thousands and thousands of changes when molecular changes when Scott went into space. And as I say, for the most part, the good news is they all return back to normal when he comes back. There was actually some immune response genes that did stay elevated, and some of them were still elevated for 6 months, but for the most part, they did come back. So the bottom line is we think these advanced technologies are going to be powerful, both for measuring future space flights. There's a number of one-year missions coming up, I'm sure you all know, and it'd be great to have these technologies going. I'd like to see more wearables done there. And I do think they're actually going to not just be powerful for them, they're going to be powerful for all of you. And I hope you all go out and get a smartwatch. And you can join our studies, by the way, including the one where you can tell when you're getting sick before you know it. All right. So the bottom line out of all this is that we've been able to do these very, very deep profiles on folks that we can follow, again, people's changes. I think I covered this enough. I want to save time for questions. I have an amazing team of people who work with me. We collaborate closely with Tracy McLaughlin, our clinical collaborator, George Weinstock on the microbiome, and the ones who led the charge are Wenyu Shao was clearly key, Kevin Contrapa and Tejas Mishra was a big part of the study, and also the space study. This personal health dashboard's been built by a team of expert informaticists. And again, there are 10 different groups. It's been an amazing group. Craig actually helped lead some of this effort as well. And I won't say all their names or I'll use up the next 40 minutes of next person's talk. So, but it's been an amazing group to work with, and I think I saved enough time for questions if anyone has any. So thanks again for having me here.
I imagine you could gain a lot of mechanistic insight if you had a way to quantify the exposome and integrate that into your outcome measures.
Great.
Are you working on that?
Yeah, could someone grab me my bag a second here? Yeah. Funny you should say that. So for the last 6.5 years, we've been measuring airborne exposures with this device. Actually, there's a precursor to this, which actually is sucking up air, measures PM 2.5, temperature, humidity, but I'll explain why I had in my backpack. I usually keep it within a few feet of me. And we've re-engineered this so it actually has a filter under it, and it captures all the particulates, including biological exposures, and under that's a chemical absorbent. So offline, then we'll measure all the biologicals you're exposed to by deep RNA and DNA sequencing, and the chemicals through mass spectrometry. And it's pretty cool. You'll discover DEET is everywhere. You'll discover that carcinogens are everywhere, but their doses will vary quite a bit. Every location is different. This is my Colorado exposure I'm measuring right now, Denver to be more specific. Because if you think about it, you don't know what you're getting exposed to right now. Nobody does, right? Nobody measures this stuff. So look offline or look it up in our paper we published about a year ago. You can see what the Colorado exposure looks like. Okay. So I do think we do need the measures, not on a city level, on a personal level. So your question's great.
Sorry, I was thinking more broadly, not just ambient air, gas environment, more broadly at the total exposure to the variety of
like food and other things as well. Yeah. So
activities, surprises in your life.
Yeah. So activity is here. Surprises. Yeah. So I wear pulse ox as well. The number one surprise is not known, but it wasn't known to me, is that your blood oxygen drops on airline flights. That's actually known in the literature. Pilots know it. Very few flight attendants know it. How many of you knew that? Probably not so many. Maybe this group, because they're. How many of you knew your blood oxygen drops on airline flights? Yeah, because you're more a space crowd, so you get it. But do you know you can get a free app called Barograph that can actually measure exactly what the air pressure is here? I'll tell you what it is. It takes a minute to count. Oh, I can tell you. It's 0.835 is the atmospheric pressure right here. And it's very sensitive if you have an iPhone or an Android. So it'll tell the difference between here and here. So you can measure exactly what's going on. 737s, Airbuses are the worst, by the way. So the point out is you can see this, and yeah, so that was kind of using what you won't see in the literature. So what does it mean? Turns out it correlates with fatigue. Turns out for me, 5% of the time, I used to be 90 or below. So there's all kinds of interesting things to learn. Anyway.
Just had a question about your immunology data, because there's been a number of studies that show the immune response decreases in space. So you get viral reactivation and things like that. Just wondered if you'd mind explaining your findings in that context.
Yeah, maybe it depends on the very specific immune markers we're looking at. There's no question we saw lots of them going up, including, and the arachidonic acid went up. There were definitely some that went down. So maybe we need to look at that in more detail. So I'd love to talk about that more afterwards. I wasn't aware of that, so thanks.
Great. Thanks.
Mike, thanks for another great tour de force talk. You're showing what we're capable of, and you're advancing that every year or every couple of seconds with your wearables. When do you think this will start transitioning into broad medical practice? You know, where the economics of doing the early work is cost effective as opposed to our, as you pointed out, our disease care network as opposed to our healthcare? Great question. So the answer is, and I'm totally conflict. I wasn't sure. I forgot to check ahead of time. I formed a company around this. I'm totally conflicted. You can disregard everything I said. That company for $3,500 will do a medical version of what I just showed you, but also do whole body MRI. And even from the first 122 people, we caught someone with early pancreatic cancer, prostate, aortic aneurysm. There's like a dozen things. It's an older bunch, I forgot to say, and that's true for our group we saw here. They're 54.3 median age entering. So the point is,
we're throwing out now. Now, why is it going to take a while to do? The whole health system's broken. We're not incentivized to keep people healthy. Nobody pays to keep you healthy. They only pay you when you get sick. So that's why people rarely go to the doctor when they're, you know, it's just not incentivized properly. So I have ideas how we should do that. I think employers should actually take a lot more responsibility. The most incentivized ones to keep their workers healthy. So that's the problem. And I think we've got to fix that whole system. It always comes down to who pays. Yeah.
Thank you.
Sorry, I didn't see the microphone here. I've left you waiting.
Oh, that's fine.
Yeah. I'll answer fast.
Okay, quick question. I wanted to get your take on companies like 23andMe.
Yeah, well, they do genotyping, simple markers. That's common disease. That's a lot less predictive. So if you're going to get your genome sequenced, yeah, get it sequenced, because the stuff like BRCA, those are they're called Mendelian diseases, single mutations that are highly penetrant. That's a lot more informative for healthcare. So go to Genos or Veritas. I have no stock in them, so they don't do a good job in interpreting, but they're better than nothing.
Yeah. Great talk. So you mentioned briefly that mitochondrial activity is different on space. I would like to ask you first how this was measured, and second, how was your opinion about the possible mitochondrial dysfunction occurring on astronauts?
Yeah, how's it measured? Well, in the end, we measured respiration, you know, using seahorse types of assays, but also by all the different biochemical markers one could see. And even Chris Mason was following actually mitochondrial transcripts and things. So there were some pretty dramatic changes. You know, there were things consistent with very low levels of hypoxia, I would argue, where it came out of some of those measurements. I'm not sure what more detail you want out of that, but we can talk more later since we're running out of time, but happy to talk. Anyway, lots of different assays were used. Happy to talk further.
I have a question about how you decide that you're healthy enough. You know, you have all this data that in some ways actually changes how you define good health. But
Totally.
Ultimately, as a matter of public health and public expense, we have to figure out what healthy enough means. And
yeah, it's a great question. I think what it means, you certainly never want to hit a symptomatic phase, right? And it's hard to know. None of us, we're all at risk for something. And so we just hope it's not impairing us or it's symptomatic. I think, you know, one angle I can add on this is we can now follow exactly how people are aging in an actionable time frame because we're measuring enough things. So for 5 measurements within 2 years, I can tell what we call your age type is, exactly how you're aging. So some people are cardioagers, some are metabolic agers, some are immunoagers. So I would argue following those trajectories is what's going to be the most important. So if you see some going off quickly, you could actually, and we have AI algorithms we're writing now to, and partially implementing, so you'll see when things are going off, and that's how you want to take action. So if you see your cardio markers going off, let's start working on that, either pharmacologically if we have to, but I would argue other interventions would be better. So I think it's more the trajectory. We capture your baseline and then watch out for those deltas, and if those deltas are changing fast, that's when you got to act. And that's how we found that one case of pancreatic cancer. That was an imaging that was changing quickly and we could zoom right in. So I think that's the way we're approaching it. Slightly different from what you might have been angling. Yeah. All right, thanks.
Okay, next up is Lucie Low, and I'd like to invite her up. She is a scientific program manager and the NIH NASA liaison. And so she brings to the table combining the basic research that's done in health and humans with NIH and applying that to what is important to NASA. And she's going to talk a little bit about some of that work. Thank you.
Thank you very much. Good morning, everyone. That was rubbish. Good morning, everyone. Yes, thank you very much. I know it's Friday. Everyone's tired. I'm still hypoxic. I'm only wearing one wearable this morning. So clearly I need to up my game, and I'm just trying to understand why I'm still so tired after a few days up at altitude. But anyway, good morning, everyone. My name is Lucie Low. I work at the National Center for Advancing Translational Sciences, which is one of the 27 institutes and centers of the NIH. the National Institutes of Health. I'm going to talk to you a little bit about microphysiological systems or tissue chips or organs on chips this morning. Talk a little bit about what they can do, what we're doing with them, and where we can go with these and why they might be useful for the space community going forward. So, oh, there we go. There's my outline of my talk. So I'm going to walk through what is a tissue chip, what we're doing, and some take-home messages. So, like I said, I work at the National Center for Advancing Translational Sciences. Our mission at NIH is to be disease agnostic. We work across multiple different diseases. We work across different processes. Our job is to transform the translational process so that treatments and cures for disease can be delivered to patients faster. We do that in a number of ways. We look for commonalities across diseases. We look for commonalities across problems. And we work collaboratively with everyone to try and really push things forward. So one of the challenges that we're currently working on, which is definitely a translational science problem, is drug development, or I should really rephrase that as therapeutic development. So over the last 15, 20 years or so, there's been an explosion in the number of disorders that have a known molecular basis, partly due to the reducing costs of genome sequencing, things like that. But still only about 500 of these, around 7,000 now, disorders actually have any kind of therapies. And partly the reason for this is because the therapeutic development pipeline is extremely costly, it's extremely lengthy, and so it can take over a decade and potentially billions of dollars to get from potential thousands of hit and lead compounds down to a clinically approved drug. So there's a number of reasons for why this huge rate of attrition is there, but one of those reasons is because the standard methods that we use to help develop drugs are not very predictive of humans. We use a lot of two-dimensional tissue cultures, and in case you haven't noticed, you are not a two-dimensional single cellular system in a petri dish. You are also not a mouse or a rabbit or an animal, which is where a lot of these other drugs go through testing. So whole animals can be extraordinarily beneficial to help us understand some of the systemic effects of various different drugs, but we're fundamentally different from certain animals, and I shall refrain from making any more jokes about how different we are from rats and things like that. But anyway, there we go. So I'm going to talk a little bit about first what a tissue chip or a microphysiological system is. So a tissue chip is essentially a bioengineered device that contains human cells in a three-dimensional structure that is designed to mimic the structure and function of your organs and your tissues. So I'll start with the one on the top left because it's the most distracting one. But this is actually a lung chip, those two little the lung alveoli and the vacuum thing next to it. So this is actually a schematic of a lung chip. So this is one of the first tissue chips that was developed. So that's a tissue chip in the little white one there. Let's see if I can make the pointer work without breaking anything. Yeah, there we are. So that one there we go. That one there is actually one of the first tissue chips that was discovered. This was a permeable membrane layer which had lung alveolar cells on one side, lung epithelial cells on the other side, and essentially the vacuum channels down the side, when the vacuum was applied, it stretches and relaxes that permeable membrane, which allows the tissues within that system to actually respond as they would with every inhalation and exhalation of your lungs as well. So it's mimicking the human lung there. I think we actually had the first video played, but if you could play it again, that would be great, thank you. So this is actually mimicking human vasculature. This is from stem cell derived endothelial cells. And those white beads you see are actually fluorescent beads flowing through the system. So showing how you can get human vasculature on a chip. If you could play the next video, please. Thank you. So this is this kind of blob that you can see gently pulsating. This is actually an innervated gut enteroid. So this is actually a self-assembling organoid type system with integration of sensory neurons, and it's actually beating and mimicking your gut peristalsis. And then if you could play the last video, please. So down on the bottom left, we have that shift from red to green. This is actually not real time, it's sped up over 24 hours, but this was liver on a chip with 20% of these liver cells actually transfected with a fluorescent reporter that changed its fluorescence profile in the presence of different molecules, in this case, reactive oxygen species. So this chip is actually a way to show us the health and function of tissues in real time over the course of a number of hours, which you just can't do in a two-dimensional system, and you certainly can't do in an animal. So the next three pictures on the top right are really showing some of the diversity of the designs of different kinds of tissue chips. They're about the size, well, they can range in anything from about the size of a USB key to something the size of a 6-well plate, 96-well plate. And these top three images are showing how They're often incorporating microfluidic channels that are providing nutrients and compounds to bioreactor type systems in the middle, where the cells are very carefully arranged in spatial and chemical gradients, so that it's really mimicking what's going on inside your body as best we can. And then those last two bottom ones on the right-hand side are actually showing integrated multi-organ systems, whether that's driven by pneumatic pumps or whether it's driven by peristaltic pumps. So the one on the left there is actually a multi-organ tissue system modeling the human female reproductive system. Is actually done by Schwo down in the front here. And that was a really nice system designed to look at female reproductive toxicity. It was able to mimic the female menstrual cycle, 28-day menstrual cycle, and also the hormonal profile of pregnancy. And the one on the right-hand side there is actually a prototype for a slide I'll show a little bit later on, which was a multi-organ system looking at doxorubicin, which is a chemotherapeutic doxorubicin-induced toxicity on a number of different organs. But the key take-home point from all of this diverse range of different designs is that all tissue chips incorporate three-dimensional aspects of their tissue modeling. They contain multiple cell types, not single cell types, so they'll incorporate immune components, they'll incorporate endothelial vascular components, as many relevant cells as they can get into the system. They all incorporate certain kinds of biomechanical forces that are designed to mimic the mechanical forces that your body is subject to with every breath you take. And as I pointed out, they can be connected. So this is not a trivial task. It's something that many of the teams are working on. But the idea is that you can put them together to create a body on a chip. So when I talk to this crowd, and I know that there are so many people who are authors of the twin study in this room, I feel like you guys have kind of peaked in terms of science because you've got your paper in Science and on the front page, and that's amazing. But we feel like we hit the big time when we got onto the front page of National Geographic. And that happened this year. Our two investigators, Clive and Sam, they're looking, I would say smug, but they were just very excited to be on the front page of National Geographic. And so honestly, that's hashtag goals for us. So we know that tissue chips are making the big time now. So just a very quick explanation of the tissue chip program at NIH. This started in late 2011, early 2012. It's now exploded, as I'll explain a little bit later. But essentially, this slide just kind of shows that all of the grants that we fund, they're 4 to 5 year projects. They're biphasic. They have, all of our teams have to meet certain milestones and criteria before they can transition to their final phase. The first phase of the program had 14 projects that were funded at the end of it. Lots of publications, that's all very nice. But one of the key points, as I mentioned before, is everything we do is collaborative. All of our teams worked very collaboratively with different pharmaceutical companies, with the FDA, who helped them design the chips and say what kinds of assays and outcomes would be helpful from a regulatory perspective. And we also collaborated very closely with DARPA, who was running a concurrent sister program to develop tissue chips and create a linked 10 organ system or a body on a chip. So that was the end of the first 5 year program. But this is my NASA swooshy slide. I don't have the same budget for a swoosh that some of you guys do, so this is the NCATS version. But this is my swoosh of where we've gone with the program. So in 2012, we had those 11 teams for chip development, 13 for cell sourcing. We began the tissue chips for drug screening program. We started the tissue chips for disease modeling program in 2016, which I'll explain a little bit more about in a minute. We also began a tissue chips in space program, which I'll also go over briefly. We started a tissue chip testing centers and database center. Now, part of our mission with this program was to develop technologies that everyone can use. So if you're creating a system in a vacuum that no one else can use in another lab, that's essentially useless. So we set up 2 independent testing centers that were charged with onboarding and validating the technology to check that what the developer says it can do can actually be done anywhere else, which is then being fed back. That information is fed back to the developer to help them create a more robust and reliable, usable technology that can be broadly rolled out. All of that information is also being fed into a database center that's publicly accessible. So all of the information, all of the assays, all of the cell information that goes into the development of these chips is actually going to be publicly accessible. We recently began a nociception, addiction, and overdose on a chip project, which is part of the HEAL initiative, which is Help End Addiction Long Term, which is a tortured acronym that I think is worthy of NASA from NIH. But this is NIH's way of trying to address the opioid crisis. And so we think there's great utility for tissue chips in understanding some of the cellular and molecular bases, not only of pain-relevant signaling, but also overdose, addiction, repeated rescue with naloxone, things like that. We have an immune chip program we've started. Incorporation of any sort of immune interaction onto a tissue chip is A, extremely difficult, but B, very, very necessary. So we've actually now set up a whole program to start working on how to incorporate not just innate immunity, but also adaptive immunity on these systems. And then as of next year, we'll be announcing some awards for what we call clinical trials on a chip. And we've deliberately put clinical trials in inverted commas like that because the FDA gets very angry when we say we're doing clinical trials on a chip. We're not actually doing clinical trials on a chip. We're using them in the context of clinical trials decision making. So for example, being able to recruit patients who are going through clinical trials, get some of their cells, create iPS-derived tissue chips from them, and then see how the responses on a tissue chip might correlate with their actual responses as they go through a clinical trial. So we see that this is going to be an additional way to validate chips as they go through this process so that we can start reducing animal use, moving straight through to human tissues through the drug development and treatment therapeutic timelines, rather than trying to do very risky first in human trials, which can be not only very risky for the participants, but also extremely expensive and lengthen that time between a promising hit compound and when it actually gets into the clinic. So we're very excited about all of these, but with the program's really exploded. We now have over 50 multidisciplinary teams and things are getting pretty busy. So I'm just going to talk a little bit about tissue chips for disease modeling and efficacy testing, which we affectionately call TC 2.0 because that was the second iteration of the first 5-year program. We have 17 teams funded through this program, 3 of them actually working on type 2 diabetes. So they'll be definitely aware of the work that Mike and his team are doing, and perhaps there's some synergies there. But I'm going to give some key take-home messages from this slide. So tissue chips, I'm going to just walk through these because I know it's early and it's Friday and etc. So I'm just going to make it very, very easy. Tissue chips can model common and rare diseases. Through that constellation of 17 different programs, we have very common diseases such as osteoarthritis. We also have very rare diseases such as hereditary hemorrhagic telangiectasia, which I like to say just because I practice saying it so much. There's great utility across a broad spectrum for not only patient stratification of different patient subgroups, but also very, very rare people, 1 in a million, 1 in 10 million kinds of diseases. As I referred to, many of these diseases can be modeled using stem cell derived sources. So the advent of induced pluripotent stem cell technology, iPSC technology, has really helped push this field forward and will continue to do so. The ability to take skin or blood from any one of you and create a tissue chip of your disease or of your own body enables us to really be able to move towards the whole precision medicine aspect that Mike was also referring to in his talk. So along with the genetic editing techniques that have enabled iPS-derived technology to be useful for these tissue chips, we also can now start being able to create or cure disease on tissue chips using gene editing techniques. So the ability to take a healthy donor blood sample, create tissue chips, and then if you know, or if you're trying to find out what kind of monogenic disorder is actually related, you can use genetic editing techniques to create that disease on a chip. You can start interrogating the molecular and cellular bases of different kinds of disorders in ways that have just never been possible before. As I mentioned, you can connect systems together. This allows you to be able to look at the systemic effects, not only of a disease pathology across the body, but also how those tissues might respond to drugs in different ways, how the metabolites of a drug can affect and alter different tissues as you move through the metabolism process. You can start looking at things like the pharmacodynamics and pharmacokinetics, that's PK/PD, of different kinds of drugs in different kinds of systems. And this is all pushing towards this concept of precision medicine, the ability to create you on a chip, your grandmother on a chip, your next door neighbor's dog on a chip. We don't do animal chips, but there are plenty of pharma companies who do because they're very interested in seeing the correlation between the animal chips that they create and the human chips that they create, which is going to enable them to see how well they tally and how much they can then reduce the use of animals during this kind of drug development process by moving straight into human tissues. So there's really a lot of utility throughout the whole understanding of disease pathologies and treatments that we see that these chips could be useful for. So I'm just going to give a couple of examples of some of the chips that have been developed through the program. So the first is an inherited pediatric cardiomyopathy called Barth syndrome from Kit Parker's team at Harvard. And essentially they are able to create a system where they take skin fibroblasts from donors, and they can create cardiac thin films, muscular thin films. So when you put cardiomyocytes onto the system that they do, the cardiomyocytes, they work together, they grow together, they create cardiac-like tissues that then when you cut the film as it's shown in the cartoon there, it actually bends up like a cantilever and it starts spontaneously twitching. Now with this twitching, you can actually quantify not only the strength of the twitch, but also how often it twitches. And so you can start seeing the function of these cardiomyocytes on these muscular thin films. Now what this team did that was really exciting is they then started comparing how these tissues from healthy donor tissues compared to those for, in this case, with this rare inherited cardiomyopathy called Barth syndrome. So you can see that on this middle panel, there's just really no twitching at all. It's not because the video stopped working, it's just because there isn't really any twitching of these muscular thin films from these patients. But what the team were able to do using genetic editing techniques is then they were able to target that specific mutation that they knew caused this disease, and using the same cells from the donor patients, they were then able to genetically rescue these thin film tissues. And so they were really able to start interrogating exactly what it is about that mutation that affects the cellular and molecular bases that cause the twitching of this heart tissue. And they were actually able to uncover a new mechanism of myopathy in this particular instance. Another one of our teams, just to highlight, this is a linked system. So this is from a team at Columbia. They have a 5-organ tissue system. They have heart, liver, bone and bone cancer, vasculature and skin in their model. And they're actually looking at, they've created a platform where they have tissue-specific medium in each chamber so that they can really enhance the development of each specific tissue with the relevant growth factors. But then they also have a recirculating common medium that is behind an endothelial barrier. So they're really trying to mimic exactly how the organs within your body are not only locally processed drugs, but also how the systemic effects of drugs might occur across the entire body. And so they're then actually looking at, as I referred to earlier, doxorubicin-induced off-target toxicity. So in their bone cancer model, they're able to add doxorubicin, which is a chemotherapeutic. It's really pretty toxic. And they're able to see the off-target effects. Now, what's really key here is that every single one of those tissues in that system can be created from the same donor. All of those different tissues are from an independent donor. Now, this is key because it not only means that each individual patient could potentially have their own body on a chip system made, but it means for things like cancer chemotherapeutics, where often some of the ways that treatment is approached nowadays is with different cocktails of chemotherapeutics. You can actually do these on a tissue chip before you start putting these into patients. You can start stratifying patient populations to see which ones are most likely to respond most effectively to different chemotherapeutic combinations. And obviously in groups of patients that are quite sick, then this is something you want to avoid, is actually trying just a bunch of different chemotherapeutics in them when you can avoid that with using these tissue chips. So we think that this is going to be a very exciting opportunity for personalized and precision medicine as we go forward too. So I'll talk a little bit about our Chips in Space program now. We're very excited to have partnered with the organization formerly known as CASIS, now the ISS National Lab, and NASA, and we sent our tissue chips to the space station. So why on earth would you do this? I feel like I'm preaching to the choir here, so I'll skim through this. So we know that there are physiological changes under even short-term and long-term microgravity. And we know that these also are very similar to some disease pathologies that we see down here on Earth, and also some of them are very similar to aging. Now, what's fascinating for us at NIH is that, as Mike pointed out, and from the twin study results, is a lot of these changes reverse when you come back down to Earth. So for NIH, this offers an unprecedented opportunity to be able to see on a very short time scale some of the molecular, genetic, and cellular changes that occur in response to microgravity that are similar to aging, but also that reverse when you come back down. So it offers whole new avenues for us to explore different kinds of disease pathologies and how different kinds of treatments might work. So we're really excited to be part of this program. If the clicker would work. Oh, I went too far now. So this is a slightly off-kilter slide. So anyone with OCD in the audience, I apologize because nothing quite lines up. But anyway, this is our program. We have 9 teams or 8 teams working on 9 projects. We have teams working on everything from gut inflammation to osteoarthritis, aging of the immune system, a couple of teams working on cardiac systems. So the key point here is that every single one of our teams is sending research to space, but working on an extremely clinically relevant problem for people down here on Earth. People have heard of kidney stones. That happens up in space, happens down here on Earth. So we're able to really try and understand some of these very well-known clinical disorders by using these tissue chip systems. Now, all of our teams have also partnered with implementation partners who have been charged with adapting their hardware for the space flight. And this is really important for us for another reason. So, like I said, everything NCATS does is designed to try and get processes and technologies and things out to make things happen faster. So part of that is that we wanted our tissue chips to be much more, we want to get them out to the community. So the Chips in Space program is really helping with this. So right now, if you're using a tissue chip in the lab, you might need something that's maybe the size of this kind of dais, or a standard cell incubator to actually house all of your chips. But then you'll need a bunch of tubes and computers, and all of the footprint and the technology that goes along with actually pumping these small chips can be quite excessive. And so, funnily enough, NASA said to us, no, you can't send 48 cubic feet into space for each one. You've got to shrink it down. So that's where our teams have been working with their implementation partners to shrink their technology down to a fraction of the size. And I know that some of the implementation partners, if they're in the room, they've had a lot of sleepless nights about trying to make this happen. So we're incredibly grateful to them for all of the hard work they did. But essentially, this has enabled all of our systems to be shrunk down to a much smaller footprint. Most of the systems are almost entirely automated with very minimal crew time intervention needed to actually keep the cells doing what they need to do. And there's downlink telemetry, so we can see often in real time what's happening with our systems. And so this is incredibly useful for us at NCATS, because it also means that now we're helping develop a really robust and reliable technology that doesn't need a skilled crew to be able to do it. Fighter pilot proof was the phrase that we were given by an astronaut. He said, hey guys, this is great, but we really don't want to screw your experiments up. So you've got to make them so straightforward that they're fighter pilot proof. And we were like, okay, your words, not ours. So we've worked to really make these as straightforward as we can so that they can be utilized across Earth in a number of different extreme environments with very limited skilled use needed. So we're very excited about some of the technological advancements from that, and some of that's detailed in this paper by Kathy Young et al. That came out recently. So we had our first 2 launches at the end of last year, and on May 4th this year, 5 of our teams have now launched. They're busy analyzing their results. We're seeing some expected results. We're seeing some exciting microgravity related changes that are currently in the process of being delved into, but we're excited to continue with our series of launches. Each of our teams has the opportunity for 2 space flights, which is very unusual. This means that they can actually build on the results from the first flight and actually incorporate those into the design of the experiments for their second flight. For us as scientists, as biomedical scientists, this is critical because science is an iterative process of failure. So the opportunity to actually have a go, to be able to build onto what you've learned for the first time and then fly again is really phenomenal. And we're very, very, very excited to have that possibility. We're also indebted to the crew up on station who did such a great job of manhandling and changing media bags and doing some of the things that needed to be done to keep our chips healthy. So we're very grateful to them. So I'll just finish with some take-home messages, and then I've got time for questions. So I think I've explained that we think tissue chips have many potential uses for things like drug safety, looking at diseases, understanding disease pathologies, looking at the different systemic effects of different drugs across multiple organs. I've pointed out that we can model disease using genetic editing and stem cell-derived technologies, which is opening up whole new doors for precision medicine, as is the ability to link multiple organs together to create body on a chip or potentially astronaut on a chip. And so this is my key final take-home point, is that we believe that these kinds of systems could have utility for space biomedicine. Now, not only would you be able to, is there the possibility of, for example, creating astronauts on a chip, where you might be able to take previous astronauts, current astronauts, future astronauts, be able to try and understand how their different organs or how their different tissues might react to different kinds of stresses, how they might react to different kinds of risk mitigation strategies, the ability to perhaps put tissue chips up on the moon and leave them there for 6 months and see what kind of effect it has from the galactic cosmic radiation. All of these kinds of risks that we know NASA is trying to address as we all move towards the Artemis missions in future. So we think that there could be some key utility there. We also think that obviously there's the ability for testing drugs, perhaps even testing drugs that have already been flown and looking at the effect of those on healthy organs and tissues down here on Earth once they return, to help try and understand how some of those effects of microgravity and radiation for long periods of time up either on low Earth orbit or in deep space could actually affect human physiological systems. So I will leave it there, and I believe we have time for questions. So thank you very much.
Great talk. I have one quick one. Are your systems prepared to have different blood, not exactly blood pressures, but to simulate the fluid shift that happens in the astronauts?
That's a great question. So our chips have been developed for things here on Earth. So that's not to say that we wouldn't be able to model that kind of fluid shift, but we haven't as yet. That said, we do have a number of chips that are looking at different kinds of cardiovascular effects. We have chips that are modeling things like atrial fibrillation, chips that are modeling the linkage of heart tissue with vasculature. So there are ways that we could start modeling those, but it has not been done yet.
I really appreciated your framework of this test lab idea, and I was wondering if you could speak more about how that's gone, how much things have been able to be reproduced.
Mm-hmm. So let me see if I still have those slides on the end here. So I had some slides on the testing centers, and I'll just hop through those. So this is actually our 2 testing centers, one of them at MIT, which is now transitioned out to a company called Javelin Biotech, and one at Texas A&M. And these teams have actually been incredibly successful at onboarding the technology. It has been an uphill struggle for them, no doubt. They found the hardest thing for them is actually getting the legal paperwork signed between 2 institutions. But once the technology transfer has actually taken place, they've been able to do, it's essentially been almost as straightforward as sending teams to the developers lab to learn how the developer does it, and then having the developers lab come to the testing centers to help them set up the systems. This is the list of platforms that they've actually set up and have been testing. And what's been really fascinating for everyone involved is that the feedback from the testing centers is able to go directly back into the developers' designs and how they continue to improve their tissue chips. So it's been really one of the key kind of scientific outcomes we've also found is that cell sourcing for these tissue chip systems is one of the biggest challenges that everyone is facing right now. I could talk for hours about the cell sourcing, but I won't because I've only got a minute left. But essentially the differences and the variability between cells across whether they're commercial suppliers or whether it's how a different lab does a different kind of differentiation protocol for their stem cell derived sources can have massive effects on the actual outcomes of these chips. So the testing centers have been really instrumental in actually crystallizing how that's a problem across the entire field and is helping come up with ideas to help address those that will help standardize some of these issues across not just the tissue chip field, but the entire bioengineering field. Okay, thank you very much.
Okay, next up, we're changing gears just a little bit, and we have Dr. HIDEKI SARUWATARI, who is from JAXA, Japanese Space Agency, and he's going to talk about the Electrostatic Levitation furnace, or ELF, and some of the work that's been done in that arena. All right, thank you very much.
Thank you. Okay, thank you, Anna-san. And before start, I'd like to thank the organized committee for inviting me to this symposium. So let's get started. Today, I would like to give you mainly three topics. One, I know maybe the audience here, more than half of audience here is a life science person, I think. So I will give you a brief introduction about the electrostatic levitation technique. And next, I will explain about a Japanese facility on board the keyboard. And finally, I would give you some results using our facility, okay? So first, I will give you our motivation why we started this work. You know, when you're handling melts or liquids, usually you need a container, okay? But if you handle high temperature melts, there are bad things like chemical reaction with container, or easy to solidify when you use a container. But if we use this containers technique, we can overcome these problems. For example, we can get precise some physical properties, and we can get large supercooling. I will describe about the supercooling more detail using below temperature timing chart. You know, when the liquid or the melt is cooled down with a container, the temperature goes down to the melting point and stays at a certain time and decreases again. But if you use the countermeasures technique, the temperature goes down below the melting point at a certain degree, maybe 100 degrees Celsius. We call this a supercooling state. And after that, there are two cases. First case is nucleation will occur, and the liquid will release latent heat to the temperature increases again to the melting point. So this phenomena is called recoalescence. Okay. Another case is the temperature goes down continuously to the vitrification. Okay. And in each cases, anyway, we can get supercooling. So if our material have a supercooling state, their structure may change as an unusual structure may have. And then the unusual structure may lead to new functional materials. So we want to get these two advantages, so we started to do this work. Okay. You know, there are some kinds of levitation technique like electromagnetic levitation uses this, and aerodynamic levitation, and electrostatic levitation. We choose this technique. This technique uses Coulomb force between the charged sample and electrodes for levitating the sample. And additionally, high-speed feedback control technique is necessary for remaining the sample to the target position. Okay. We started to develop this technique for more than 20 years, and during the term, we got some achievement using ground-based levitators. I will introduce some example for achievement. Okay, example one is this. You can see the periodical table for a metal, and the height of the each bar means melting temperature, you know, and color means level of achievement. Purple is levitation and melting, and blue color means density measurement. Green color means density, surface tension, and viscosity. And pink color means also density, viscosity, and surface tension measurement. But these values obtained only by our ground-based levitator, and for the first time in the world. Okay. And second example is shows effectiveness of precise some physical properties. These shapes means turbine blade casting simulation. Okay, so a certain company told us to measure the viscosity of their material, and before we getting the value, they used maybe a written value or a literature value. Thank you very much. Written value or literature value, and the simulation results shows no shrinkage. But after we measure the viscosity and uses these values, the simulation results changed dramatically. Shrinkage appears. So the company could know the necessity of manufacturing condition have to be changed before they started the manufacturing. So this is so powerful, I think. And next, third example is an example for the producing new function. Okay, this is a barium titanate. Barium titanate is used for capacitor for electrocircuit, and this graph means the electric constant of this barium titanate. So the left one is the electric constant obtained using normal process, means a container process. The highest values is about 4,000. Okay, but if the barium titanate was processed using a containerless process technique, levitation technique, the value is 30 times larger than the usual values. So for this material, we got a new function using a containerless technique. Okay. So even if we got some achievement using a levitation technique on ground, we wanted to our levitator to be on board the ISS because there are so many advantages in microgravity. There are many things, but the most important is this. On ground, we need force against the gravity. So for oxides, they do not have, because they do not have so much charge on surface, it is very difficult to levitate on ground. But, you know, there are no gravity, so very easy levitation can be achieved in microgravity. So this is our motivation for sending our facility to ISS. Okay. So this is our facility. ISS Earth is installed in multi-purpose small payload rack 2, MSPR2. And this is a rare one. And our facility can deal with a sample of 2 millimeter size diameter. And more than 15 samples will be processed without cool time. Okay. And the facility equips 4 heating laser, and the temperature of sample will be measured by pyrometer. And atmosphere is about maximum 2 atm or low vacuum. And you can get a density, surface tension, or viscosity using this facility. And the ISS Earth consists of mainly 4 parts: left part, center part, and right part, and sample cartridge. And main body is in the center part. And center part, main body is a 24 hedral shape, and there are many, many sensors, lasers, cameras are on it. And the sample samples are installed in sample holder. 15 samples are installed in sample holder, and the sample holder will be attached to the sample cartridge, and a crew insert the sample cartridge to the ISS Earth. And when experiment, the sample will be pushed out by upper rod to the center point between the electrodes. These are the electrodes. And after experiment, the sample will be returned to the sample holder by a Coulomb force and a lower rod. Okay. And these are the images of 3 cameras. You can see the sample using these 3 cameras. The left upper one is a overview camera. You can see the samples overall movement by this overview camera. And the right one is a magnified camera. And by using this image, you can get density. You can calculate density using these images. And the left below one is the image of a pyrometer. Pyrometer equips also a camera. And you can see here the temperature of this sample. It shows more than 1,200 degrees Celsius. So next, this slide describes the density calculation method. You can, from the magnified camera image and using image analysis and mass of return sample, you can calculate density and time profile. And you can have the temperature time profile using a pyrometer. And we use this and this to get our density temperature profile. Okay. And this, for measuring a surface tension and viscosity measurement, we use oscillating drop technique. And a helium neon laser will be illuminated to the sample, and the shadow area of the sample is detected by power meter. And sinusoidal wave shape voltage is superimposed to the sample. And when this case, the sample oscillating, so the shadowing area of shadowing will change, and power meter detecting these changes to give a vibration timing chart, and to Fourier transform, we can get also a frequency chart. And decay time and peak frequency, we use peak frequency and decay time to get surface tension and viscosity using these equations. So there are many things since we launched our facility. Okay. We installed our facility in January 2016, and initial functional test started, but it continued so long time. During that time, many troubleshoot was necessary. This is me. And to solve these problems, we did so many software update or hardware update. And finally, initial functional test was finished in March 2018. But also after that, the success probability of levitation or melting was not so high. But after the last summer's software hardware update, the success probability increased a certain level, maybe a satisfying level. So this slide shows recent on-orbit experiment results. Here, this sample is alumina thrown into the center point. These are electrodes. Okay, and soon will be laser will be on. A laser on, and you can see here the temperature. Maybe difficult to see, but it goes up to the 2000 degrees Celsius very soon. So the cause, reason of the movement, we don't know yet, but maybe the laser force or the charge change, maybe. Okay. And not yet melted, but soon will be melted. Please wait. It takes time. Soon. Yeah, aluminum melted. You can see here the perfect sphere. It means melted. Okay. And next we get surface tension and viscosity using oscillation dropping. Okay. Please see this section. Sinusoidal wave will be imposed. And you can see here the time decay here and frequency peak here to get viscosity and surface tension. And experiment finished, to laser off. You can see here the recoalescence soon. Yeah, that was a recoalescence evidence. And after experiment, the sample will be returned to the sample holder by Coulomb force, very fast, very fast. It means this sample had relatively high charges, so very fast. From now, I will show you some results of our facility. Mainly is a technical demonstration experiment. First is density of molten alumina. Left chart means temperature timing chart, and right chart is a density temperature chart. The red dot means our data, and black means other researchers' data. For more alumina is relatively easy to be levitated on ground. So there are few ground-based data. So we can compare our data to the others' data to know the consistency. And we think this difference is not so high. And we think our capability, ISS's density measurement capability is verified. And this is measurement results of gadolinium oxide. Right graph means density temperature chart. And vertical line, this vertical line means a melting point about 2,700 Kelvin, very high. But we can get the data. And this is a world first data, so no reference. And this is also for the kind of rare earth oxide, alluvium oxide, also the very high temperature point. Okay. And this is a graph for our so far achievement of rare earth oxide density measurement. Black one is alluvium oxide, red one is a gadolinium oxide, blue one is a holmium oxide, and green one for the terbium oxide. Okay. And this data is not updated using real mass because we don't get these real sample on ground. These are still on ISS. After we get the real sample, we will update these values using mass of the samples. Okay. This graph shows surface tension and viscosity of molten alumina. Left one is surface tension, and right one is viscosity. And red dot means the value of ground-based levitator, and black is the data obtained from ISS Earth. And we think this data shows a consistency. So also the surface tension and viscosity measurement capability is verified. Okay. So this video is, I will show you as a review. Okay. This is easy to understand for overall our experiment. Earth is installed in keyboard. Their advantages, their abilities. sample cartridge, sample holder. You can put 15 samples here. Set sample to the cartridge and install to the main body. Levitation by Coulomb force and melting by 4 lasers. Yeah, melt here, melt now. Density measurement and drop oscillation. Observed with IVS, Intelligent Vision System. It's a kind of camera. Time decay and frequency peak. Laser off to solidification. Very strange shape. Means maybe a solidification expansion. The outcomes we expected. Okay. And we just studied normal operations. So many projects, missions are waiting for us. Very long line, including NASA round robin. This is a project of Professor Douglas Matheson's. Okay. And summary, we developed ISS ELF, and using ELF, some physical properties, high temperature melt was obtained, successfully measured. And we are conducting various experiments using our facility. And we welcome new users. If you are interested in our facility, please send email to this. Thank you very much for attention. So any questions?
Thank you very much for sharing your thermophysical properties adventures. Do you have sufficiently adiabatic conditions to measure heat capacity?
Capacity, not yet. Yeah.
Thank you.
Quick question. When you're making your drop oscillation measurements and backing out surface tension, is there a way, did you foresee a possibility in future you'll be able to actually get a surface viscosity in case there's oxidated layer or anything on the surface? And interfacial viscosity, do you think your resolution will have enough data, high enough resolution to be able to back out an interfacial viscosity?
You mean two-phase materials?
Not a two-phase, no, surface viscosity at the interface of the drop. Instead of the bulk, viscosity in the bulk, also viscosity on the interface itself in case there's oxidation on the surface.
You mean internal viscosity or?
Viscosity on the surface of the drop. Could be.
Surface. Or we think we can measure the viscosity of, you mean viscosity distribution?
Dilatational or shear viscosity at the interface.
I don't know.
Too far ahead.
I don't know.
Great, thanks.
All right, thank you very much.
Thank you.
Sorry. Next up, we have Dr. Don Hassler, and he's going to take us a little bit off planet now and look at some rover data. He is the Science Program Director at SWIRI for Southwest Research Institute and is kind of local. So thank you very much.
Thank you, Annalisa. Thank you, everyone. So I'm going to talk a little bit about the MSL RAD instrument. RAD stands for Radiation Assessment Detector, and it's a HUMD funded instrument on the SMD funded Mars Science Laboratory, which is currently operating on Mars. I'm going to talk about it from the point of view of a space weather outpost on Mars. Fundamentally, initially, RAD was funded to characterize the radiation environment on Mars as if it were a single measurement, and you get it, you're done, and you move forward. The truth is, it's much more complicated than that there's time variation of all things. And these days, people are talking about space weather as kind of encompassing the radiation environment as well as other factors. But I think the new challenge that we're facing when we talk about Gateway and other exploration to the moon and to Mars is dealing with interplanetary space weather, and how do we monitor it, how do we predict for it, and how do we protect our astronauts for it. So, as I mentioned, the key element of space weather is space radiation. Space radiation is different than radiation that we normally experience on Earth. It's higher energy in general, but it's composed of two types. Galactic cosmic rays, which come from outside the galaxy, from supernova remnants and explosions. They're very high energy, but they're very kind of relatively low intensity, but they are modulated by the sun. They're modulated by the solar cycle and slow variation by the heliosphere. And then there's lower energy, but sometimes higher intensity solar energetic particles, which are directly from the sun. And they occur sporadically and generally around solar maximum, but they basically are lower energy than the galactic cosmic rays. So on Mars is actually unique compared to, say, the Earth, for example, in the sense that Mars experiences directly space weather and the space radiation environment for two reasons. It doesn't have a global magnetic field like the Earth does, and the atmosphere is much thinner. And so on the surface of Mars experiences space weather directly. So I mentioned galactic cosmic rays. They vary slowly with the 11-year solar cycle. You can think of that as the sunspot cycle, but it's basically the active regions on the sun. The sun gets more active every 11 years. It's kind of a rough gauge. It's not always 11 years, and it's not, and as I'll mention later on, it's not very uniform. And solar energetic particles are big storms. They're coronal mass ejections. They're the things that cause the aurora, that knock out our satellites occasionally. You hear about those, and they're important for all aspects of, well, when we have astronauts on space station, when they go on spacewalk, we need to make sure that the sun is quiet during that time. So on Mars is an additional complication in the sense that the primary radiation gets fractionated in the atmosphere. And so you actually have, say, a single particle at the top of the atmosphere, and it breaks into 1,000 different particles by the time it gets to the surface. And so to validate your transport models, you really need to characterize and measure all those radiation types. Also, from an astronaut safety point of view, protons and helium are not particularly damaging to astronauts, but the higher energy particles like iron and carbon and nitrogen and oxygen, those are. And so you need to really measure all of those different particle types to really understand the danger of deep space radiation for astronauts. So RAD, here's a picture of it in the upper right. It's basically the size of a microwave. No, not even, the size of a blender. It's about 1.6 kilograms, and it's, you can see sitting in the rover on the right, the window of RAD facing up, but that's location on the rover. And it's basically characterizing these two types of radiation, galactic cosmic rays and solar energetic particles. It takes about 4 watts when it's operating, but it's It's quite smart. It's autonomous, so it turns itself on. It takes data, and then it goes back to sleep so that we manage the energy budget on the rover. It's also can be, we're actually flying one on space station now as well. It can be used as an area, it's being used on space station as an area detector. You put it in the different rooms and you can characterize the radiation environment in each of the different rooms or regions of space station, as well as the different angles that it's looking. It basically measures both charged particles and neutral particles all at the same time. Actually, I need to be using this one. So basically, RAD characterizes, it measures protons all the way up to iron. It does energetic particle spectra of a few MeV per nucleon, from about 10 MeV up to over 100 MeV per nucleon, as well as neutrons from a few MeV up to over 100 MeV for neutrons, as well as gamma rays, and of course LET and dose equivalent measurements. Here's a picture of RAD being installed into the rover. It's actually inserted from the underbelly, but that gives you kind of a size with respect to humans installing it onto the rover. So it's interesting, about a few months before launch, we realized that we could turn RAD on during cruise, and we could actually learn what it would be like for the radiation environment for an astronaut inside a spacecraft on their way to Mars in deep space, or something similar to it, a proxy to it. And so we convinced the project to allow us to turn RAD on, and we turned RAD on basically a couple of weeks after launch, and we got about 6 months of data during cruise on the way to Mars, even though it was designed to be a measurement on the surface of Mars. So this plot here shows galactic cosmic rays, which is this low energy, kind of insidious, not really background, but a component which doesn't vary very strongly. You can see lots of dips and variations from the basic structure of the heliosphere as it goes through. But then we also had about 4 or 5 solar particle events, or these solar storms. So both of the two types of radiation that I talked about were being observed during cruise. So we learned quite a bit during cruise from those measurements. So basically, if you added up the dose rates from those solar particle events, this line right here, and compared it to your total dose or dose equivalent from the cruise phase, we found out that about 5% of the contribution to the total dose equivalent during this 6-month cruise phase was due to solar energetic particles. Well, that's a highly variable number. It's not always the same. Sometimes it's closer to 0. Sometimes it could be an order of magnitude more. And I'll talk more about how that varies in a minute. On the surface of Mars, actually, it was really it was amazing. The morning we landed on Mars on the 6th of August. The first instruments to be turned on were the imagers, of course, but RAD was the second instrument to be turned on. And the morning that we were getting ready to turn RAD on, we were reading the New York Times, and it turns out that the turn on of RAD, characterizing the radiation environment on the surface of Mars, on another planet, was exactly 100 years to the day, the anniversary of the measurement of the first energetic particles on Earth by Victor Hess with his high altitude balloon experiments in Germany. So it was fortuitous, it was serendipitous that here we were 100 years after discovering galactic cosmic rays and energetic particles, we're actually measuring them on the surface of another planet. So here's a picture of RAD on the surface of Mars with the scenic backdrop of Gale Crater. And we've been operating successfully for the last little over 7 years now. We're slowly working our way up the valleys and canyons of Gale Crater up to, up to as high as we can get. We'll see. I'm not sure we're going to make it to the top, but it's still an adventure. So this is basically the dose rate. It's just a simple number to visualize, similar to the one I showed in the previous slide during cruise, of the RAD data on the surface. So you can see a few solar particle events. You can see a relatively large one here. You can see it going up and down, varying, lots of dips, which I'll talk about in a minute. And then right around here in 2015, we started to, this was relatively, well, we had a pretty weak solar maximum, but it was solar maximum nonetheless. This is solar maximum. And then we started to decrease, the solar activity started to decrease, and we started to decline into a solar minimum. We're close to the the bottom of solar minimum. We don't know exactly. It could turn around in 6 months. It could turn around in a year and a half. Last solar minimum was exceptionally long, and we didn't expect it to be so long, so we really don't know when this will happen. But we had this one large event, actually right kind of in the smack dab middle of, you know, descending phase of the solar cycle, close to solar minimum. These dips are called Forbush decreases. They're basically when the heliosphere acts sort of like a cloud, and it kind of blocks the galactic cosmic rays from reaching the surface. It's called a Forbush decrease, and we see a lot of those. It actually takes a relatively high energy particle to make it all the way to the surface. So we've only really observed a few solar particle events on the surface. This is really a pretty wimpy solar maximum, kind of disappointing from a science point of view. But we did get this one relatively large event, which then I'm gonna talk about next. So on September 10th of 2017, we had actually a solar particle event on that one that I showed the spike for, both on Mars and at Earth. So we were really excited because this is the first time we've ever observed a solar storm on 2 planets at the same time. And actually they were almost opposite each other. It was also at the same time of a Hungry Ghost Festival. So there are parts of the world, they call it the Hungry Ghost Festival event. But what's interesting is the sun was really approaching solar minimum. There was this small active region going across the sun. We were tracking it. No one really expected anything to happen. Here's the orientation. Here's Earth. This is Mars over here on the opposite side. And this is kind of a sketch of this large storm that erupted. I think the next slide will show it in video. No, the next slide maybe shows it in video form. Yeah. So this is the eruption of this event. This is a model that actually happens after the fact. You know, obviously you need to get the data points to process where this event is. But it shows that, you know, it wasn't direct hit on either the Earth or Mars. It was kind of an oblique angle hit on both planets, but it was observed on both planets. And what's fascinating is it caused an aurora on both planets. It caused an aurora on Earth, which we see a fair number of, quite a few, especially if you live up in Alaska and Norway. But it also caused an aurora on Mars, which is the first time we've observed that. That's observed with the Maven spacecraft in orbit around Mars. It also produced solar particle events both on the Earth and Mars. And this is actually the RAD data, which I'm going to talk more about, but this is the observations from GOES, which show an increase in the solar energetic particles. But it was really also, there's a thing called a ground level event, which is a neutron monitor term, which is historically how we measured these large events. And it produced quite a bit of, it produced a ground level event, which hadn't been seen in half a dozen years on Earth because it had been such a weak solar cycle. But it also produced a ground level event both on Earth and Mars. So how big was it really? It seemed like it was big. We were kind of starved for activity, so it was the biggest one we've seen. How big was it really? These are the data from CRUISE. This is the data from the surface, which I just showed. So let's go back and recap we had from CRUISE. We had about 25 millisieverts of dose equivalent from the combination of all 4 solar particle events during CRUISE. If we look at the data on the surface, it was really only about a millisievert of dose equivalent contribution from this. But it's interesting when you start to look at this and we start to look at the data, there's a thing called the quality factor, which is what you use. It talks about the bioeffectiveness of energetic particles, how effective they are in doing damage to biological tissues. And then the higher energy particles are higher Q. So galactic cosmic rays have very large Q, solar energetic particles have small q. So even though the intensity was large, it actually blocked some of the galactic cosmic radiation. And so there was a decrease, a dilution of the Q factor from the solar particle event, which in retrospect, it kind of makes sense, and all discoveries make sense in retrospect, but it was a discovery nonetheless. And so we had this decrease in quality factor, which kind of minimized the impact of this event. So let's study the event a little bit in more detail. This is the quiet time galactic cosmic ray level on the surface. This is the the initiation or onset of the solar particle event on Mars due to the dose rate from this storm that happened, and then it starts to decrease. And then there's a point right here, which is when that cloud that I mentioned, the Forbush decrease hits, and it actually blocks the energetic particles getting to the surface of Mars. So it actually, the result of this Forbush decrease reduced the level significantly, so that the post-event level of galactic cosmic rays was actually lower than the pre-event level, because the heliosphere had been disturbed. There was these, this coronal mass ejection basically filled the space in the heliosphere, and it basically reduced the post-event levels. And so when you look at it kind of overall, we had this large contribution in dose equivalent due to the event, but then this reduction after the event. And so the net increase from this was not large. It was a small increase in the total dose equivalent if you took, say, a 1-month average or so. So in summary of what we observed, we saw the dose rate increased by a factor of 2 to the event, and about a factor of roughly 2 times increase in dose rate due to what we'd seen previously. However, the reduction in the Q factor and this Forbush decrease made it kind of it was a wash. Now, however, this is just one event. Maybe it was just a coincidence that it was just the right energy with a Forbush decrease. A lot of people suspect that the Forbush decrease has a maximum effect, and so there are potentially other events that could be much larger than this, which you'd still get the same reduction from the Forbush decrease and the reduction of the dilution factor, but a much higher level from your event. It's one event. We have a single data point. You really can't do statistics on one event. So how does this compare with other historically large events? This is really what we want to know. When we want to send astronauts into space, we need to, be prepared for the worst case scenarios. And in the energetic particle world, the worst case scenario is called a Carrington event, or a superstorm. And it's basically, arguably maybe 10 times worse than the things that we normally observe. The last Carrington event was, well, we haven't really been measuring these types of measurements since about the space age, which is 20, 30 years. But there were Carrington events speculated in 1890 and in the 1950s, but we have much more information now. But we can compare this event to what we have before. These are the rad observations during cruise from the galactic cosmic rays. This is the rad observations of the solar particle events during cruise from all 4 of them. This is the combination of the solar energetic particle events just on the surface of Mars, which is much less. Now, if you take the modeling of these historically large solar particle events, 1956, 1959, 1960, 1972, 1989. 1989 was probably the largest event that we've seen in recent history. That's the one that knocked out the electric grid in northeastern United States and Quebec. It's talked about a lot. The Space Weather Prediction Center and SRAG all talk about this as the example of, maybe not the worst case example, but certainly an example why we need to be paying attention to these things and monitoring them in real time. But the point here is that any one of those large events could be much, much larger than what we measured on the surface. We just haven't seen it yet. We had a weak solar maximum. Who knows what the next solar maximum will bring? It'll be in the next 20 years. So what are the prospects for the next solar cycle? Well, the solar cycle is very difficult to predict. These are all sorts of different predictions from the best and brightest we have, and they vary. Some people say we're going to have a very large solar maximum. Others say we're going to have a weak solar maximum. And the timing of the solar maximum is highly variable as well. We had the deepest solar minimum of the solar age, of the space age in 2009, and it wasn't predicted to be that way. So cycle 24 occurred late and was weak. We don't know. The next cycle could be potentially weaker and longer. We really don't know. The other thing that's interesting is that solar particle event in September was this declining phase of the solar cycle. It's actually interesting. Many of your events happen when the magnetic field is most complicated, so declining phase or ascending phase of the solar cycle. But what we see is that we get solar particle events and coronal mass ejections at all times in the solar cycle, not just solar maximum, the ascending phase or the descending phase. So we really need to be monitoring this as well. And there's other effects. I'm talking about radiation here, but there's other, the solar maximum, which is worse from the point of view of energetic particles and coronal mass ejections and acute events, is the opposite of what you get. Arguably solar minimum might be the worst time to send astronauts to Mars because you get an increased solar galactic cosmic ray levels. And so, you get increased solar particle events, but decreased galactic cosmic rays or vice versa. So characterizing the extreme events is something that we need to continue to do. We're learning a lot that the radiation environment on Mars is just not a single number. It's a time varying number, and it's integrally related to the solar cycle. The Space Weather Prediction Center and SRAG, the Space Radiation Analysis Group, which supports NASA's human exploration endeavors. They've been taking data and collecting observations and monitoring for 50 years. Space Weather and Space Environment Center or Space Weather Prediction Center is focused primarily for Earth assets and low Earth orbiting satellites. SRAG is primarily for human astronauts, and human exploration. But taking the long-term data sets and observing these things over time is really necessary to understand these extreme conditions, whether they be solar particle events as extreme conditions or highs of galactic cosmic ray levels during solar minimum for extreme conditions. The other thing we're learning with RAD is that having data sets of different regions or different longitudes around the sun so that we can study them and understand them, really our predictive capability. I mentioned it, but I really didn't go into it in detail. Our predictive capability for these events is pretty weak right now. We really don't understand what it is that causes them to erupt. We're pretty good at nowcasting them when they're happening. We pretty much know they're happening, but we're not very good at predicting when they happen. And we don't know the extent to which they expand out into the heliosphere or how high of energy they will be when they do. So studying more events from multiple locations, whether it be GOES in Earth orbit or space station or RAD on Mars, which may be on the other side of the sun at the time, I think is one of the key things that we need to do. So this type of endeavor, I think, is important, and it's all part of this context of the space weather monitoring endeavor that we're proceeding with as we move forward. So thank you very much, and I'd like to make a huge thanks to Gail Allen and to Chris Moore and to Jason Cruzan at AES and HUMD in general for their support over the years for RAD. Thank you.
Hi, that was a great talk. I love this work. So the data that you've been collecting on the surface of Mars, you were showing the graph of the dose rate on a daily basis. Have you been also breaking that down by the types of particles and energies that are coming in?
Absolutely. And the problem is to talk about that in detail is a whole nother talk. And but yes, we have spectra for every single one of those days. Actually, for every data point that we have a full spectrum. I'm sorry? Oh yes, yeah. We also, and I only had 25 minutes, so there's only so much you can talk about. We could talk about this more after if you like. But we're characterizing the neutron environment on the surface of Mars as well. And for astronauts, when we go there, it's extremely important because the neutron albedo, it's the interaction of the galactic cosmic rays with the regolith. And so whether they're direct neutrons coming from the atmosphere or albedo neutrons coming from the regolith, they're important. And so if we're going to build a habitat on the surface using regolith materials, we need to understand those measurements. And so yes, we're characterizing that as well. Thank you very much.
All right, thank you very much. One more hand for all of our speakers, please.